Hey everyone, I hope you're doing well in this isolation period. My name's Ed, I'm a final year PhD student at Cardiff University, and today I'm going to be talking to you about my project, which is the genomic basis of preservative resistance. Hopefully I'll be bringing something new and exciting to you today. Failing that, it'll just be new, and failing that, at least the weather's nice. Thanks for tuning in anyway. So just to kick things off, why would you care about preservative resistance? So when we think of antimicrobial resistance in bacteria, which even in the face of a pandemic is an ongoing global crisis, we tend to think of antibiotic use in the clinic. However, the manufacturing industry is actually one of the largest users of antimicrobial agents in the form of preservatives. Now, these are used to preserve home and personal care products. These are products mainly based on oils and proteins, so they are substrates that can be readily used by bacteria. Now, preservatives exert an antimicrobial effect and prevent growth in these products, which includes soaps, shampoos, and conditioners. However, many organisms that we encounter are what we call intrinsically preservative tolerant, some of which cross over with those found in the clinic. Examples include Burkholderia, Pseudomonas, and others. Now, at the moment, we faced a additional crisis in that we have to reduce the environmental impacts by moving towards green and minimal formulations, both in terms of the environment, but also the consumer. Now, there's an example of a preservative system known as the isothiazolinones, try and say that 3 times quickly, which a few years back started causing skin reactions in some patients and consumers. It's therefore important that we move towards these minimal formulations to reduce the impact on the consumer, but we also need to maintain adequate preservation. Maintaining adequate preservation by using a more minimal system requires full understanding of preservative resistance, and unfortunately, at the moment, we don't actually understand why it's happening. And essentially, this is where my project comes in. Now, at Cardiff, we've been working with Unilever for a number of years to tackle why preservative resistance happens in bacteria that are tolerant to preservatives. Now, the best way you can get information out of bacteria, as any good bioinformatician or molecular biologist will tell you, is by looking at the genome. Now, the genome is defined as a complete set of genes or genetic material present in a cellular organism. And what this basically means is that if we get the complete sequence of a genome of a bacteria, it can tell us a lot of things about it. Now, the first part of my project was looking at what the predominant organisms are that we might want to look at. We've got a lot of bacteria at Cardiff that we've isolated over a number of years, but it's important to hone in on the important ones. Now, there's a lot of information on these pie charts here, but what I want you to focus on is panel B, looking at the non-food product recalls. Now, this information comes from something now known as the EU Safety Gate database, previously called RAPEX database, and this is essentially a government catalogue of all recalled products, along with the organisms that contaminate those products when they're reported as a recall. And what you can see here is that a large proportion of organisms remain unidentified in these products. This is an entirely separate issue that I won't touch on today, but you're welcome to follow up this publication listed at the top of the slide if you want to read more about it. But beyond that, predominantly what we see are gram-negatives. Now, gram-negative bacteria that we mostly see are Pseudomonas aeruginosa, which is a problematic clinical pathogen, particularly in immunocompromised patients, and also the Enterobacteriaceae. So what we went on to do was sequence a bunch of these organisms, and what we found is that preservative tolerant bacteria, i.e. those that we have isolated from industrial backgrounds over a number of years, possess megaplasmids that make them unique. So if I can refer you to the figure on the right-hand side of the slide, you'll see that I've shown a genome of bacteria. Now, these 3 circles represent all the DNA in the bacteria. DNA in bacteria typically tends to be circularized, not always, but as a whole, it tends to be. And what you'll see here is that this industrial Pseudomonas aeruginosa genome that I posted, RW109, has 3 circles that we refer to as replicons. This consists of a main chromosome, a megaplasmid, and a large plasmid. Now, the reason this is particularly unique is typically in Pseudomonas aeruginosa, you would see the main chromosome of an organism, but this megaplasmid and large plasmid wouldn't exist. So just to show how much of the genome these megaplasmids constitute, if you had a Pseudomonas aeruginosa genome that had, as I've listed here, a 7 megabase pair main chromosome, an additional 550 kilobase pairs due to a megaplasmid, you're already nearing a tenth of the genome size, so 7,700,000 base pairs that are accounted for by plasmids, due to the fact this megaplasmid is 550,000 base pairs, the large plasmid is 150,000 base pairs. So you can clearly see here that there's been a massive increase in genome size just due to the presence of these plasmids. So I'll keep using the word plasmids, but what are plasmids and why are they relevant to what we're talking about? So plasmids are circular DNA molecules that can be transferred between bacteria and replicate independently of the main genetic content of the bacteria. The reason they're a problem is that they can pass on any genes between bacteria in a process known as conjugation. Now, plasmids usually will range anything from about 3,000 base pairs in size up to about 20,000 base pairs in size. And as the name of the megaplasmid suggests, these plasmids are much, much larger. Why is this a problem? This basically means that megaplasmids have a much larger repertoire of genes that they're able to pass between bacteria. Now, if we took an example from a clinical setting where we have two bacteria, one has a plasmid and one has a megaplasmid that is acquired from a donor, they both have antimicrobial resistance genes in the plasmid. The one with the megaplasmids potentially has more antimicrobial resistance genes as a whole, just due to the size of the megaplasmid. Now, interestingly as well, we've sequenced a number of bacteria, and if you remember on the previous slides, I mentioned that the predominant organisms we find from recall products are Pseudomonas and Enterobacteriaceae. And in both of these organisms, in a number of studies that we conducted, which you are welcome to follow up afterwards, we have found megaplasmids. So there's clearly a trend here between preservative-tolerant organisms and the possession of megaplasmids. The key message to take away from this is the size of megaplasmids that it can pass on many more resistance genes than plasmids. And as a final note, what are the consequences of megaplasmids and how long do they stick around? Now, I've described plasmids as things that are passed around quite regularly, but if they're passed around, do they stick around in bacteria, or are they passed on and do they change? Now, what we're looking at here is something called a core gene phylogenetic analysis, and essentially what this looks at is the evolutionary relationship between different bacteria. Now, anything that you see on this diagram here that has a label next to it in a straight line means that the organisms are very, very closely related. The more branching there is in the phylogenetic tree, the more evolutionary diverse these organisms are. And the key ones that I want you to focus on this diagram towards the bottom are the bottom three blue labels, RW130, 146, and 131. So these were three isolates which were isolated from the same manufacturing background over 6 years. And what you can see here is that the megaplasmids of these organisms are identical in terms of core gene content. Now, we know from the literature that megaplasmids are highly flexible because they're passed between different bacteria and they can actually recombine with the main chromosomes of bacteria and freely change homologous regions of DNA. What you can see here is that over the same 6-year period, all of these isolates, despite the fact they've been identified as the same thing, have maintained identical core gene content. Now, if you consider the flexibility that a 550 kilobase pair plasmid offers, there is a lot of room for genes to be passed on, changed over a number of years as the organism requires it. But what's interesting is that these three isolates have not only maintained the plasmids, they've kept the same core genes. This seems to suggest that the plasmid confers some tolerance of the background that they were isolated from. This is something we're currently following up via plasmid knockouts and other experiments, and we hope to report more on this shortly. This has been a bit of a whistle-stop tour over what has been a difficult but interesting 4-year project. Thank you all for listening, and I'd be happy to take any questions now.